Unit 11, Lesson 3, Complex Punnett Squares, aka Dihybrid Crosses. First things first, our lesson objective. So what are you learning about today? You're going to learn about a student should be able to predict outcomes of dihybrid crosses using more complex Punnett squares. So we did the very simple ones that are just 4 boxes. Well, today we're going to do ones that are 16 boxes. So the warm-up question is, in the wild, Beyonce fruits grow in clusters of either 3 or 4. Clusters of 3 are found in organisms that are homozygous recessive for this trait. Assuming that the fruit growth patterns is controlled by this single gene, what would the Punnett square look like if we're breeding a heterozygous plant with a homozygous dominant plant? So to answer this question, you need to know what homozygous means, what heterozygous means, what recessive means, and what dominant means. Then you're going to set up your Punnett square. Okay. Homozygous dominant, heterozygous. Remember, homo means same, so these are both big, or these are both going to be the same, and then it says homozygous dominant, so they're both going to be capital, they're both going to be big. And heterozygous just means that they're different, so this is one allele and that's a different allele. All right, then we're going to fill in our Punnett squares. So what do you think is going to go here? Well, the way we figure that out, remember, is we just look up here, X, big X, over here, big X. How about for this box right here? How do we figure that out? Well, little x and big X. And you repeat it for all 4 squares. When you're done with that, this is what it should look like. Now, back to the question. The question asks, will any of the offspring have clusters of 3? How do you know? Well, remember, in the word problem, it said clusters of 3 is the homozygous recessive trait. It's a recessive trait. Are any of these recessive? No. This is heterozygous, heterozygous, so the dominant is going to win. And these are both homozygous dominant. So there's no recessive offspring, which means none of the offspring will have clusters of 3. Okay, so just reviewing monohybrid crosses, let's do some word problems. So cystic fibrosis is a hereditary disease that affects the respiratory and digestive systems. Cystic fibrosis occurs when 2 recessive genes, little c, are present. A person with 1 allele for cystic fibrosis is called a carrier, big C, little c, of the disease. If the mother is a carrier of the disease and the father is homozygous dominant, what are the chances that the child will be a carrier of cystic fibrosis? There's a lot of information here. What I like to do is I like to annotate. I like to break it down. So first things first, let's find out the parental genotypes. We know we're going to have to do a Punnett square, so we need to know what are the parents' genotypes. All right, so the mother is a carrier of the disease. So a person with 1 allele for cystic fibrosis is a carrier. So the mom's genotype is going to be big C, little c. How about the father? What is the father's genotype? Well, he's homozygous. Homo means same. Homo means same, and they're dominant. So we're going to have big C, big C. Boom. Just by reading the problem, you figure that out. Now, the question is asking, what are the chances that the child will be a carrier of cystic fibrosis? So what genotype are we looking for? We're looking for big C, little c in the offspring. First things first, we're going to set up our Punnett square, and then we're going to look for if any of the offspring will be a carrier. So we put our parental genotypes here, and now we're going to fill in each box. So in this box right here, we're going to have big C, big C. Okay, which of these Punnett squares is done correctly? Well, we know right here this box is wrong because there's a big C up here, but there's no big C in the box, which means this Punnett square is wrong. This one is correct. Okay, so again, the question is, what are the chances that the child will be a carrier of cystic fibrosis? So what are the chances of the child being a carrier? Okay, and as it said, the problem told us carrier is big C, little c. So 1, 2, so 2 out of 4, that is 50%. Okay. If you're still struggling with simple Punnett squares and word problems, you definitely need to reach out to me and let me know, because we're about to do dihybrid crosses, which are way bigger and whatnot. Okay. So yeah, monohybrid crosses, you're going to read the problem and determine the parental genotypes. You're going to set up your square and determine the offspring. After that, you're going to look back at the question. Look at the question. What is the question asking you for? And then find that information out on the Punnett square. All right, another problem. Another practice problem. So the characteristic for curly hair is dominant to the characteristic for straight hair. A cross between two individuals for hair type is shown in the Punnett square below. What must the genotype of the parents be to produce the results shown in the Punnett square? So this is a little bit different. Usually, we've been doing problems where we gave you the parent genotypes and asked you to find the child. Well, now we can do it another way and give you the child genotypes and ask you for the parents. But it's the same exact concept, just backwards. So, right here. Big C, big C. Boom. Right here. Big C, little c. Boom. You can double check. Big C, little c. Big C, big C. Okay, that's all you do. All right, so that's what what the offspring told us the parents had to have been. So, is this hetero or homozygous? And is this hetero or homozygous? Well, homo means same. These are the same, so that's going to be homozygous. This one up here is going to be heterozygous. However, look at the problem. It doesn't just say homozygous. That says it's homozygous dominant or homozygous recessive. So, or what are these? These are dominant. So we have homozygous dominant and heterozygous parents. So there's multiple ways we can ask you the same kind of concept and whatnot. Okay, now let's get into dihybrid crosses. Punnett squares where we're looking at 2 traits at a time. So dihybrid cross is, as everything is in biology, we have we can break down the word. Dihybrid cross. So slice that word in half, or not in half. Slice that word into its separate parts. Di means 2. Hybrid means a combination. Okay, so we're looking at 2 things combined, and we're predicting their probability. Whereas before, we would just look at 1 trait. So it would be like, we look at leg length. Either the parents' legs were short or tall. Now we're looking at what are the probabilities of 2 traits being inherited together. All right, so a basic example. Let's start off with a basic example, and then you're going to have some practice problems on your own. So what are the possible genetic combinations among offspring of a man who is heterozygous for both dimples and freckles, and a woman who is recessive for dimples and freckles? Okay, so we're looking at our offspring. What combination of dimples and freckles will there be? Well, the first step is going to be write down the parental genotypes, as always. Write down the parental genotypes. What is heterozygous? What is recessive for dimples and freckles? So for dimples, let's just keep it simple. We'll just make this D. And freckles, we'll keep it simple and make it F. So dad is heterozygous for dimples. So big D, little d. And he's heterozygous for freckles. So freckles is going to be big F, little f. Boom. That's dad's genotype. Now how about mom? Mom's genotype is recessive for dimples, so little d, and recessive for freckles, so little f, little f. Boom. Now what do we do? Now we're going to try and set up our Punnett square. But before we set up our Punnett square, we have to do something else. we have to determine all possible gamete combinations for each parent. Since we're looking at 2 traits, we have to find out all the different combinations that one parent can make, and then look at the probability of parents together. So you do that by doing this thing that's called FOIL. I believe in math class you guys use FOIL as well. FOIL, the first set, then we have the outer set, then we have the inner set, and then we have the last set. So FOIL, first, outside, inside, last. If you are confused on that, pause this video right now and look at this picture and take your finger and do like that. And now you can see A and B are first, or big A and big B are first, right? Then the outer set. So then we have big A and little b are outside, right? You notice how they're outside. We have the inner set, the inside ones, and then we have what's last. Okay. This is another little animated GIF. First, outside, inside, last. And they're just showing this is dad and that is mom. They're showing it all together. Oh. Well, they've already shown moms. Okay, so basically with moms, the first FD. The outside, little f, little d. Insides, little f, little d. And of course, the last, little f, little d. So mom's gametes are all going to be little f, little d, little f, little d, little f, little d. How about dads? Dads might be a little bit more confusing. So let's take it step by step. First, so the first one. So we have F, big D. Okay. Outer, big F, little d. Inside, last. Sorry, I had a brain. Okay. FOIL. So now, this is all the possible combinations of alleles in dad's gametes. All the possible combinations for mom. Now we're going to use these to set up our Punnett square. So, Punnett square is big. Much bigger now. Now it is 16 boxes. So 4 by 4. Going to put our dad's gametes that we found out using FOIL, and we're going to put mom's gametes that we found out also using FOIL. Then we're going to do like we've always done. We're going to figure it out. Go box by box. Right here. For the F's. Little f, big F. Little d, big D. So in this box, we're going to put, right, this, that, and this. Boom. Next box, right here. It's going to be those and those. The next box, right here, it's going to be those and those. And so forth for the entire rest of the thing. Same thing we've been doing. As long as you set up your parental gametes, you do FOIL correctly, then doing this part is super easy because you're just writing them down. You're just copying it right there. Now, this is all the different offspring genotypes that there could be. Okay, so I have some questions for you. Now that we've taken our parental genotypes, we found our parental gametes, we put them on the Punnett square, figured out the Punnett square, now we can finally answer our questions. So, question here is asking, which percentage of offspring can be predicted to have freckles, so that's going to be a dominant, and dimples? So who's going to have both freckles and dimples? So let's see, right here. They have freckles, they have dimples. That's 1. They have freckles, they have dimples. That's 2. They have freckles, they have dimples, that's 3. They have freckles, they have dimples, that's 4. They have freckles, oh, no dimples. Freckles, yes, dimples, no. Freckles, yes, dimples, no. Freckles, yes, dimples, no. Okay, how about down here? Freckles, no, no freckles. They have dimples. No freckles, but they have dimples. And same thing here. How about down here at this row? Freckles, dimples, neither. So, for this specific Punnett square, the only offspring that are going to have both freckles and have dimples are going to be these 4 up here. So we have 1, 2, 3, 4. 4 offspring out of the 16 are going to end up with freckles and dimples. 4 out of 16, if you have a calculator, 0.25 or 25%. All right, what percentage of these offspring are predicted to have freckles but no dimples? So we're looking for freckles, big F, and we're looking for no dimples, little d. So freckles, we already know those have freckles and dimples, so that's not what we're looking for. We're looking for freckles with no dimples. So that's going to be these 4 right here. Boom. Okay, one thing that I want to note is that the way that this problem worked out is that they are all in a row. But not every problem is going to be like that. Not every problem is going to be like that. Some problems are going to show you different things. Okay, just so you know. All right, so step 1 is to determine the genotypes of both parents using the word problem. Step 2, you're going to use FOIL, so first, outside, inside, and last, to find mom's gametes. Then you're going to use FOIL again to find dad's gametes. You're going to set up the Punnett square with the gametes from mom and from dad. Okay, so the parental gametes. Then you're going to fill in the Punnett square to determine the potential offspring. Then, using the word problem, you're going to figure out the phenotypes of the offspring. All right, and that is the end of this lesson. Now you have a bunch of practice problems where you're going to spend time really making sure that you have this concept in your brain. I have your tests right here, and there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 Punnett squares problems on there, and I think, yeah, half and half are dihybrid. Some are dihybrid and some are monohybrid. So definitely use your time wisely and use the raise your hand feature if you have any questions for me. Okay.